Okay, I want to introduce today the co-founders of a really cool platform startup called Integrated Biosciences. We have Felix here as CEO, and co-founder Max is joining as well. I won't steal their thunder. I will just sort of say that Project 8P is starting to embrace partnerships and collaborations with companies, with startups that have technologies that we think are well suited to investigations in 8P. And so from time to time, we'll be inviting such startups to tell us about their technology, and hopefully we will be able to report on actual data in the months ahead. So with that, Max, Felix, why don't you guys introduce yourselves and your company, and then the stage is yours.
Great. Thank you for that, Ethan. So by way of introduction, I'm joined here by Max Wilson, a professor of molecular biology at UCSB. And as you guys can see, my name is Felix. I run Integrated as their CEO, basically. And what we do is really, we started Integrated around a year ago with the premise that we can target age-related, but also in general, stress-related kind of pathological cells in a more systematic manner. So with that, I'm happy to kind of share with you guys the stuff that we're doing and also the road ahead for us in terms of what we think we can do for our AP heroes. So in terms of the organization, we were told that we have around half an hour. So what I'd like to do is to share around 15 minutes, just giving everyone on this call a really high level overview of our company, our milestones, what we've been able to accomplish as, again, this 1.5-year-old new biotech coming from labs at Harvard, MIT, et cetera. And then in the second half of this call, led by Max, we'll kind of delve a bit more deeply into the biology and what we think of as really exciting approaches that, again, we can create value as well as maybe create new therapies for our AP heroes. So, Max, do you want to say a few words before I delve in?
No, I'm really grateful to have your attention today. Go for it, Felix.
Great. Yeah. So again, I'll be sharing a few slides, but if there's any kind of questions on the call, feel free to just chime in. It doesn't have to always be a structured presentation. But again, we founded Integrated in 2022 with the goal of curing age-related diseases. And in many cases, what we found was really something that, Max and I reflected on really day to day, which is really that we're not living longer, we're dying longer. This is very salient in what we see in very aged people, for instance, the 80, 90-year-olds that, of course, we all know in our daily lives. They're not really living healthily, typically. Of course, there are exceptions, but in many cases, we see things like neurodegeneration, very, very kind of different age-related diseases. I mean, I can just go down a list of like metabolic disorders, et cetera. But the main idea is that a lot of people suffer from age-related diseases. And in many cases, why these age-related diseases occur is really because of biological stress. So what the past 20 years of biological research have shown is that this idea of cellular stress drives aging. And we focus on aging as really a catch-all for really what we think of as really chronic but debilitating diseases. The main idea being that what I'm saying when I refer to aging also refers to diseases and also kind of conditions like, for instance, AP and other neurodegenerative or kind of debilitating, really, diseases. And so I guess to really, land a punchline here is that what we decided to start Integrated on was really this observation that cellular stress underlies aging. And also there are no drugs that target these very fundamental principles of cellular stress that drive aging. Okay. So at Integrated, what we're doing is developing clinical assets to target these very fundamental cellular stress responses. And the analogy that I like to make is that we are treating sources of this universal and conserved biological stress in the same way that antibiotics treat bacterial infections, meaning that we go after specific small molecules we go after acute indications, and then we try to cure kind of these acute indications with powerful small molecules that lead to binary outcomes. In the case of our AP heroes, what we envision is that our small molecules that we've already discovered will have binary effects. For instance, will it cure AP, for instance? Okay, so that's kind of the high-level overview. Our company can really kind of do this line of work because we are the first ones that have really developed a proprietary optogenetics platform that virtually mimics cellular stress. That's kind of a lot of, technical terms that I just threw in there, but the main idea is that we have synthetic control of aging. We can use synthetic biology to control aging and other stress-related pathways, such as the ones that are relevant to our AP heroes. Okay, and we've combined this new approach of being able to synthetically control biology with unprecedented chemical compound libraries. And together, these two factors are integrated really by a proprietary AI platform that enables us to create the world's most predictive AI models for drug discovery. And when I say that, I really mean it. We have the world's most predictive AI models for drug discovery. By way of example, these technologies have already led to more precise drug candidates faster than our competitors. This is demonstrated by our rapid discovery of the first novel class of antibiotics, one of the few in over 60 years. And for comparison, the last such discovery took all of industry over 35 years to accomplish. We just published this in Nature just a few months ago. And what we have done here is to compress the discovery timeline from decades to months. But of course, we are comfortable publishing antibiotics as a demo, really, because antibiotics, from a company-wise kind of perspective, are not profitable. It's also certainly hard to develop. And I would say there's huge unmet need, but perhaps even more dire need for other diseases like age-related disease, as well as, finding cures for our AP heroes. So what we as a company focus on, we focus on kind of developing the equivalent of antibiotics, but for universally needed drugs, such as ones that go after aging and such as ones that go after, conditions of cell stress, for instance, those related to our AP heroes. So we've also really led the field in developing these AI approaches to anti-aging small molecule drug discovery. And that's just what I've shown here as kind of evidence of the technical progress as a company that we've been able to put out. So to give you a sense of what we've already done in this company, in this space, our company really has been around for just over a year and a half. And with just 5 full-time employees, we've developed 5 lead compounds. Our typical programs test over 100 times the number of compounds that pharmaceutical drug companies screen, both experimentally and in silico. And our assets, several of which are shown here, we have some assets that are pre-IND, and in general, they target age-related diseases with significant unmet need and universal market potential. Okay, and I'd like to kind of say a few words about AP in particular, but to give you a sense for who is leading the charge at our company, right? The magic really happens here because our founders are literally the best people to take all of the latest advances in AI and apply them to small molecule drug discovery. So in particular, something that I like to highlight is that our founding team are all physicists turned biologists who currently run hardcore experimental biology research programs. So we don't hide behind like code. We really like pipette all day, okay? And together with Jim, who also co-founded the Wyss Institute at Harvard, as well as the field of synthetic biology, we really see this once in a career opportunity to make Integrated the preeminent company that deals with cellular stress responses as well as aging. So what we really envision, and this is something that we'd love to kind of share in a bit more technical detail in the coming minutes, is a collaboration with Project AP and our AP heroes, in which we apply a few things. One is our currently developed small molecule assets. They target very different sources of cellular stress that, as I mentioned, are universal. And there is significant scientific evidence to believe that when you have substantial, say, rearrangements in your DNA, such as those that are experienced by our AP heroes, that leads to a cascade of cellular stress responses that basically fall straight in the purview of the cellular stress responses that we've been kind of targeting as a company for the past year and a half. So what we already have are small molecules that can modulate, that can control these cellular stress responses in different ways. And one of the key aims of our proposed collaboration is to really test what we currently have and see if any of these small molecule assets, again, that we're currently developing for other diseases, might also work to be disease modifying for our AP heroes. To start, we'll kind of think about this in an in vitro manner in which we have in which we will leverage the resources of Project AP and also additional scientific resources to see if in a test tube, any of our small molecules might be disease modifying, with the eventual idea that we can eventually develop these small molecules if they were disease modifying for applications in AP. A different kind of orthogonal approach to all of this is that, of course, we've been taking a hypothesis-guided approach to targeting specific cellular stress responses. But of course, AP is a complex phenotype. And so what we are also kind of envisioning is a collaboration in which the second arm, really what we do is to identify new targets through comprehensive sequencing and screening approaches that, again, we as a company have developed. So of course we have our assets, but of course the assets may or may not work. We also are kind of looking forward to looking at AP in a more hypothesis agnostic way and identifying new pathways and new targets to drug. And the culmination of all of this work will eventually lead to both new drug candidates for our AP heroes, as well as basic scientific findings that can guide new development, not just in terms of drug discovery for us, but also moving the AP field forward. So with that, would love to take any questions. The audience has been a bit quiet, but feel free to chime in. Otherwise, we can definitely go more technical if there's no additional questions.
Yeah, and I'll talk a little bit more about the science in the second half.
Are there any families that have questions about like what this means?
Don't be shy.
I think someone's muted. I think Faye is trying to talk?
Oh yes, Faye, you're on mute.
Oh, sorry about that. I clicked the wrong button. It sounds absolutely fantastic. I think my only question so far is really about, you showed the sort of 5 programs. Would they all be addressing oxidative stress? And Maxwell, if you're going to talk about this next, then please just let's launch into what you're going to say.
I can mention a little bit about this and then kind of add more color to it. So basically, it turns out that we have a number of conserved stress responses in our cells. And interestingly, many of them are sort of agnostic to the kind of stress that the cell is experiencing. So like, for example, one of the ones I'll talk about in a sec, the same exact response is invoked to deal with viral infection or like starvation, right? And so it actually is why we think that the molecules we're developing might be really powerful, especially for something that might induce pleiotropic stresses like a chromosomal abnormality. Yeah.
Great, thank you.
And I guess to also add a bit more detail to what Max has said, right? Like that was a highly technical exploration, basically saying that our stress responses will deal with different stresses, including oxidative stress. Okay, so our assets are kind of, in a sense, a bit more fundamental than just dealing with specific stressors. And certainly, I would say around 2 or 3 of our 5 assets deal with pathways that respond to oxidative stress.
Thank you.
And you used a phrase earlier when you were talking about small molecules being disease modifying. High level, what does that mean? The disease modifying portion of it?
Yeah, great question. Basil, is that correct? So sorry if I'm
It's okay. It's Basil, but
Basil.
I've been called a lot of worse things.
Yeah, so the main idea of disease modification is that we have drugs, small molecules in our case, that actually go to targeting the sources of disease, the molecular or cellular kind of sources of disease. And this is as opposed to things like cold medicines, which are really symptom alleviating, etc. Of course, the idea of targeting disease at its source really is a stronger modality than being symptom alleviating, because we're hoping to make cellular level modifications that ultimately cure the disease.
It's like the difference between chemotherapy for cancer and morphine for cancer, right? Like both of them might ultimately make you feel better.
Thank you.
All right, Max, go for it.
All right. Let's make sure. Okay. Can you see the right
I think
slide?
you center mode, maybe, if you want to go to full screen.
Swap display. Does that work now?
Yep.
All right, cool. Okay, so yeah, so I thought I'd dive into the science and the biology a little bit more. This is, I understand that we have some non-scientists here, so I really pride myself in being able to communicate clearly. So please stop me and let me know if I can kind of unfold any concept for you. Today I'm going to talk about this notion that we have a variety of cellular stress responses, and how basically we've built tools to virtually control those stress responses. So meaning we can turn them on or turn them off in the absence of real stress, and how that allows us to discover the small molecules that we're now trying to bring into the clinic. Okay. The pathway we'll focus on today is called the integrated stress response, and it actually is what gives our company our name, and it's also its name comes from this idea which you brought up, Faye, which is that multiple different stresses are actually sensed by the same response. So that's why it's called integrated. And I think by virtue of it being so core to our cells feeling either stressed or unstressed, it is actually affiliated with so many important cellular decisions and disease states. Okay, so just thinking about the typical kind of life cycle, if you will, of a cell when it's stressed, is it first is unstressed, and then it goes into this stress state, and there this stress response gets turned on. And this stress response actually has some options in our bodies in order to deal with that stress. So one thing that it commonly does is it can tell the cells that they need to kill themselves. They need to commit apoptosis for the betterment of the organism as a whole. And that's a fine strategy, because we're made up of hundreds of billions of cells. If the stress is not too high, then actually it creates these adaptive mechanisms that actually restore the cell back into its normal state, what we call homeostasis. And that is sort of unlocking those mechanisms and being able to drug those mechanisms might be the reason, give us ways of actually kind of rejuvenating cells that we're looking into. One of the kind of fascinating things is that there's this other third state called chronic stress, which definitely is, I think, likely going on in the patients with chromosomal disorders, and but also it's affiliated with a huge number of other diseases. So this chronic stress is affiliated with diabetes, many, many neurodegenerative diseases, including all the major ones, Alzheimer's and Parkinson's, and general things like cognitive dysfunction. And maybe some of you can notice that many of these disease states actually occur at a higher frequency in people as they age with chromosomal aberrations. So the other kind of very interesting thing here is there's one more state. When the cell gets stressed and it comes back to this homeostatic state, it actually is stronger. So this is called hormesis, and it's this notion of what doesn't kill you makes you stronger. This is a real biological notion, and it's sort of why working out, doing things like sauna and cold plunge actually make you like healthier and live longer, or associated with health and longer life. So we wanted to study this. This is one of our company employees, Tyvon, who is heading this. And so we thought, and we've been thinking very deeply about why the ISR might be activated in these chromosomal disorders. So here, there isn't as much work in chromosome 8, but certainly in trisomy, there was a really landmark paper for us, which showed that if you were to stop the cells from sensing stress in these mice, that actually the cognitive dysfunction and the memory impairment is alleviated, completely goes away. And we have actually sort of, we're pioneers in the cell biology of these responses, and we've actually shown, this is extremely technical, I know, but that the stress sensors of these in these cells actually go and touch some of the other sort of RNA, they're sort of organelles that deal with dysregulated macromolecules, which are likely abnormal in these chromosomal diseases. And for the aficionados, this is the P-body, which is sort of the mRNA processing body, which and since chromosomal abnormalities likely lead to large changes in the amount of mRNA. And so I think that there's a really good chance here that we are on the exact right target for these types of diseases. So it's been typically traditionally very tricky to work with these diseases, or sorry, work with these pathways and try to find new drugs that target them. And that's because we have been unable to find mechanisms that allow us to control the cellular stress response in the absence of stress. So prior to us coming around, people would put small molecule poisons on cells, like this one dapsigargan, which comes from like some sort of like organism in the ocean, and that just damages the cell. And this damage is irreversible. It activates multiple cellular stress response pathways, so it doesn't allow us to do like on-target screening. And actually, it ultimately just causes the cells to die. And so we asked whether we could uncouple the damage from the activation of the stress response pathway. And this was the key to unlocking our small molecule screening platform here. So what we did, I think I'll skip over. What we did is we actually looked at the design of the stress sensors in the cells, and we actually found that we could engineer them to respond to light in the absence of stress. So think of like when you put on virtual reality goggles and I simulate that you're on a roller coaster. You're not actually on a roller coaster, but you experience that you're on a roller coaster in just by shining the right wavelengths of light at the right time at your eyes. We're literally doing the exact analogous thing to cells, just with sort of a molecular virtual reality headset. So this is sort of the scientific, fun part of how the tool works. Basically, we take these proteins from plants and we cluster the receptors, and that causes us to activate them virtually. I think I'll probably just skip to this slide here. So we're able to show that this works. We have, followed up and have been looking at many other stress sensor pathways. But I think one of the key kind of aspects of this technology is that we really, for the first time, have virtual control of these cells. And so we can, for example, virtually activate a very specific sensor, like, for example, the one that we hypothesize might be the most strongly associated with memory deficits in chromosomal abnormalities. And then what we can do is we can then look for small molecules that modulate the sort of internal workings of the cell and read out the biology at the outcome to see if we're finding molecules that modulate the biology in the presence of the virtual stress. And so by having these sort of three pieces, the ability to virtually activate the stress response, the ability to look through large libraries of small molecules of potential drugs, and the ability to, in a fast and efficient way, read out the biology, we can kind of sift through molecules that we think are very promising for specifically targeting these cellular stress responses. So I think I'll sort of
So
pause.
can I ask a quick question?
Yes.
So when you mean virtually, you mean because you don't have the damage, but you're actually doing the experiment in the cells. You're just
Yes.
activating it. Okay. Because when I think of virtual, I think of it's not real, but
Right.
what's not real about it is the damage. You're just
Exactly.
activating. Okay. So it's still an actual experiment in addition cells.
Exactly. And in fact, just to put that into perspective for you, so in just under a year, we use these 384 well plates to do our little screen. So they have 384 little experiments in a plate, and they're about a centimeter thick. And if you stack them end on end, they would be taller than the Salesforce Tower in San Francisco. If you looked at the ones we've done just in this past year.
And what are the readouts? So, when you activate this, obviously you're looking at some downstream process and you're seeing that changes when you add the molecules. Is it different for every protein that you're targeting, or is it a similar response that you're looking for in your plates?
Yeah, so let me show you. I can show you a bit more about that if you're interested. Let's see about this. So here's like an example of a kind of experiment we do. So we like deliver all this light input, and we kind of can fix the cells, and sort of a question. And then these are images of the cells, and they're stained for all these various markers of the stress response. So for example, this one on the left, bottom left, phospho-EF2 alpha, is sort of canonically associated with inhibition of translation, and translation is essential for the formation of new memories and refreshing and maintenance of synapses. So when EF2 alpha is phosphorylated, it's highly associated with neurodegenerative diseases. And so we can just simply look at it with stains, antibody stains. And as you can see, like we're kind of modulating it here based on how much light, the intensity and the duration of the light we're giving. But we have sort of a huge toolkit of potential readouts here. So other things in the pathway, we can look at cell death. We can also, look at the whole transcriptome, so we can do very high dimensional readouts. It just depends the exact nature of the question we're asking.
And then these three type of profiles. So I guess the one on the left is you get a rapid onset followed by some attenuation or versus the one where you seem to blow out the signal kind of the whole way. And then the other one, which is kind of this time and dose response. Are these kind of generally the three types of qualitative outcomes you see when you interrogate these markers?
Yeah, at this time scale. Yeah, basically like these three responses account for like 80% of the variance of the genome of the transcriptome that we see. Yeah. And what's interesting is these like kind of pulsatile responses, these are associated with adaptive mechanisms, so like autophagy, protein refolding, and these sort of slow linear increases are associated with terminal outcomes, so cell death. So in fact, one thing we noticed is that if you kind of overlay these, you can see that they're already turning on the cell death mechanisms even as they're kind of trying to adapt. So cells are kind of preparing to die even while they're trying to fight the insult. Yeah. Great.
So Max, in this, is this generally saying that, say, in a disease where there's a damaged protein created, that it's more the stress responses around the presence of that and mechanisms that created it, rather than its effect directly on biology?
Yeah, I think that's one of the key hypotheses here. So there are open questions, just to be clear. So maybe the stress response is actually actually it's certainly causal of many of the phenotypes we see, but maybe it's like maybe that's the compromise that biology is willing to make for not letting the damaged protein create more other worse damage. But I think that there's very promising mouse evidence right now that shows that we actually can turn off the stress response in some of these models, and the mice seem fine, their memory enhances, they're more active, and there don't appear to be sort of long-term problems. So it's very promising. Yeah.
Okay, so fascinating. So in this case, you might see, in terms of a disease modifying, you'd see reduction of symptom rather than a full curative component on, say, a gene therapy that might one day repair the DNA itself and the resulting proteins.
Yes. Yeah, exactly. And but I would say there are two things I would say to that also. There might be key moments in, let's say, brain development where if you inhibited these responses, you could sort of allow a lot more, let's say, neurogenesis or other things that are being stopped to occur, and like sort of get people through sort of a high damage part of the disease. So that's one kind of interesting possibility we've discussed with Ethan, and one thing we can potentially screen for. And then the other kind of thing is there might be kind of drug like regimes where you cycle on and cycle off. So you let the stress response do its thing, then you turn it off and you let your cells refresh all the proteins at the synapses, and then you stop and you let the stress response sort of do its thing again. So I think there's a lot of kind of flexibility here too, in terms of how to manipulate the response with these drugs to maybe do better than what we have.
To add one more thought to what Max just said, it is also quite difficult to use, say, gene editing to directly, say, cure like AP and other. These are large DNA rearrangements.
Yeah.
And by drugging the phenotype directly, our hope is that we can, again, be disease modifying and target the cellular kind of level generation of this phenotype. Sorry, Ethan?
Yeah, I was just going to get at the question of like, ultimately, targeting this pathway, does that require sort of intermittent dosing or continuous dosing? I know that's getting ahead of things, but have you guys given thought to, in AP in particular, which regime you might prefer?
Yeah, I can take this. So that's a great question, Ethan. And so what we currently have is data showing that our lead compounds that modulate the stress response are not cytotoxic. So, or have really any medicinal chemistry liabilities of the ones that we've tested for. And up to date, we've tested for quite a lot. So we're quite confident that if needed, the compounds could be dosed in severe ways and also in kind of repeated ways. But that being said, there have been prior mouse models showing that you can dose very intermittently in kind of a hit and run kind of fashion, and you can get a lot of therapeutic benefits. For instance, with previous small molecules that were discovered to also modulate the integrated stress response. So there's a lot, I would say, a lot of leeway into kind of figuring out that question.
Yeah, it reminds me of the questions about rapamycin for health span, like weekly dosing at this amount versus daily dosing at this smaller amount, and what's sort of the optimum strategy.
Right.
Then
we obviously have a lot of different phenotypes within our community, right? Inversions and deletions and duplications and trisomy, and they all kind of they're different in lots of different ways and similar in some. So assuming that this will be looking at things across the full population too.
Right. So actually that's exactly the question we had. And that's one of the first things we're doing is we're getting cell lines that represent sort of the full gamut of different reorganizations. And we're going to do just basic characterization on these lines to see if they have the same stress response abnormalities, or if there are ways we can break them down into subgroups.
Well, that's interesting. So you may see actually different subgroups within the population have different stress response signals based on
And so like just to throw a hypothesis out there, like if I think duplications might show something different than just deletions, just because duplications, you literally have twice as much mRNA that might be overloading the sort of macromolecular processing machinery. Deletions might be something different, right?
Nicoletta?
Hello, thank you for that. I don't know if it's time for questions or you had more things to present, because I can wait till the end.
No, we can just talk about the question.
So we have done some initial characterization on the stress responses in 8P fibroblasts, and we had 4 lines of them. And we looked at ER stress and mitochondrial stress and integrated stress markers at transcriptional level. And we did not see any increase in those. On the contrary, BIP is lower, so ER stress is lower in 8P. Mitochondrial stress, it's lower. And for integrated stress response, CHOP is lower at transcriptional level, and ATF4, ATF5 are a little bit more spread around, so it's not statistically significant. But the cell lines individually, they respond quite differently.
Right.
We have also done some characterization of oxidative stress with mitosocs. We looked at mitochondria more specifically, and it matters that the cells are under a state of stress to see something in the cells, not as baseline. So we use treatments to see that there is more increase in 8P rather than in control. And also in terms of mitochondrial function, the cells show more differences when they are in galactose rather than glucose medium. So there will be a few things to consider. One is the variability of the cells. The other one is that maybe the cells that we are using are actually not showing high integrated stress. Maybe that needs to be induced. And why do they not show high integrated stress? Because all the theory says that they should be showing that. Mm-hmm. So I think there are a few questions to be looked at. We also did phospho A to alpha, but with antibody immunofluorescence, but I cannot recall anything dramatic coming up. So that's another thing that is probably looking.
Yeah,
So,
thanks for that, Nicoletta. I guess it would be fascinating to kind of delve more deeply into the biology. And actually, one of the things that Max and I also see for cells that we have from disease populations, so these are, say, neurons from people with Alzheimer's, et cetera, is that the ISR, the activation and whatever kind of biomarkers one looks at might also be dynamic, meaning that they, as you mentioned, Nicoletta, they might change in response to things like environmental conditions, as well as even different timing, like when you look at it under, say, the microscope, that might also change. So there's that, and also a confluence of additional stress responses. So Max mentioned the integrated stress response as really one of the stress responses in which we are looking to do a lot of detailed characterization. But of course, we have, even if the ISR is not, say, the stress response to target in this case, we are also targeting other stress responses, including senescence.
And then I add two other things. So we do have a cool experiment. We can dynamically profile the cells. So in the presence of this virtual stress. So we can see, like, for example, if certain cell lines amplify or like suppress stress in interesting ways. And then we have compounds that can sort of restore the dynamical profile as well. And so, we've actually seen some of, I guess, some of this RNA-seq data that showed that, CHOP and some of these downstream markers are lower. And we actually have produced a variety of what we're calling small molecule potentiators, where they actually make the cells respond more robustly to stress. So perhaps those are going to be useful if you just take the data at face value there.
thank you.
I got one more. Sorry if I'm dominating the time.
No, go for
it. I tend to do this. I got to jump to another call for work, so I just wanted to get it out. Is there any, as outcomes are concerned in some of these models that you're doing? And I know, early intervention is a big deal, neuroplasticity and all the things that go along with that. Is there anything to when this type of thing gets started in your models, if it's early or late, does it make a difference in outcomes?
Just to be clear, you're talking about like treating people young?
Yeah, young, like young mice, early or whatever models, or what's the expectations, I guess, in that, if we're not there yet, right?
Right. I mean, I think sort of the general rule is that interventions early are always more effective, right? Just because of the loss of plasticity, let's say, of the tissue. And so I think we would like to have these, ideally we would like to have these compounds working, in people as young as possible, right? I think the other kind of interesting scientifically, or the other kind of scientific variable here is we see that a lot of these age-related diseases are aggravated, like the signal gets more and more as these individuals age, right? So, when you think of a clinical trial, if you need to have measurable outcomes, and if there aren't great measurable outcomes at the molecular level when they're younger, that could be a challenge that we'd have to think about. But I've actually heard some pretty interesting ideas from Ethan on this front.
Well, I'd love others to jump in too. I mean, I guess I'm an eternal optimist, maybe an insufferable optimist, but let's assume that we get, let's say your tool compounds, there's something really exciting in terms of phenotypic normalization. What model would we go to next? And, I mean, set aside the whole question of what clinical endpoints you look for in a human being, how would you get conviction in terms of efficacy models beyond in vitro that we found something that could be useful? And then, of course, the question of when you intervene matters. But I guess, yeah, have you given thought, or has anyone given thought to, if we did get promising in vitro correction, what would we do next to actually give us more conviction to ultimately go into humans?
Yeah, that's a great question, and I can take this one because basically this is quite relevant for what we're doing as a company, which is we have also been working on partnerships with, for example, the Alzheimer's Drug Discovery Foundation to also put the same compounds or optimized analogs thereof into things like Alzheimer's mice, Parkinson's mice, etc. So what we envision for our collaboration with Project AP is assuming that the in vitro results all support it, we would eventually like to start preclinical testing of these compounds or, again, optimized variants thereof, also in mouse models. Of course, the caveat here is that in general, when I say mouse models, what we're thinking of are general mouse models of neurodegeneration, things like, again, Alzheimer's mice, things like concussed mice, etc. Just because, at least for AP, it is, I would say that in general, it's not well studied, but perhaps more so for AP, how mouse models and animal models might reflect human disease etiology. But from a high level, that's kind of our approach and our thinking towards the subject.
Right, because we obviously don't have a bona fide mouse model per se. I think in conversations with Harui, maybe a Down syndrome mouse, given the convergence we're seeing in NPCs, maybe a Down syndrome mouse is like a poor man's AP mouse for now. But obviously we need to pressure test those assumptions. But I guess that was kind of what you're saying?
Yeah, exactly. I think if we can develop new mouse models, and of course, we have developed new mouse models for things like looking, for instance, at P16 mice and like senescence, for instance. But that would be one of the capabilities that we look forward to. But of course, given what's currently available, I think it's fair to say that we would have a lot of confidence in our compounds for disease-modifying AP if it works across a range of neurodegenerative models. And that's really because we're targeting these core conserved stress response phenotypes that should, if our hypothesis of targeting the phenotype is disease modifying is correct, should truly be disease modifying across different diseases.
I mean, ideally, we somehow assess neurodevelopment in that model too, not just neurodegeneration, which obviously there are fears that this is what's looming. But right now, the acute day-to-day concerns are around the neurodevelopmental presentation.
Yeah, we agree completely. And one of the things that we also have baking in terms of other projects is we are also running, say, very large-scale mouse experiments where we keep track of not just things like development, but also things like transcriptomic profiles across different organs across time. So we will eventually be able to generate very high-quality, detailed data that might give us more handle points for seeing if the compound works eventually for humans in AP heroes.
All right, anyone else want to jump in? It's kind of, we're open season here for questions.
I can't find the hand thing, so I'm just doing it. I'm just going to shout out. So I think this is brilliant. This is really exciting stuff. Can you tell me then, and sorry if I've sort of not understood properly, you sort of showed the three examples there, the IF2 alpha, the ATF4, and the CHOP. So is that the sort of like the three main molecules from, or proteins from one particular disease profile? Is that like the top three essentially from like essentially diabetes, so something like that? And then with a different disease, you'd see a different three. Is that how this is looking? Or maybe CHOP would come up in diabetes and also Alzheimer's. Am I getting this right?
So basically, those three are they tell us information about how that stress response is being activated.
Okay.
They basically are the acute. They're each one is related to biological processes. So it allows us
you're
to
using these as the measurement.
What's that?
You're using these as the measurement, is that it? Like to sort of measure the stress response?
We're using them as
Yes.
biomarkers.
Yeah.
Okay.
There is so one tells us the translational state of the cell, so how much protein it's making. One tells us how much of an adaptive response it's trying to do. And
Okay.
then final one, CHOP, tells us how much the cell's trying to kill itself, basically.
Sure. If you're headed to apoptosis or not. Gotcha.
Yeah.
Okay, so that's what your readout is essentially from every experiment.
That can be one of them. We can have, we can go much higher dimensional if we need to, or if we want to screen 400,000 compounds, we could do something different based on imaging or, we have a lot of options, I guess.
I see. Okay, thank you. I understand that a lot better now. It's still very exciting.
And just to kind of double click on one point, right? Like, I think that your idea of kind of associating specific diseases with specific kind of these biomarkers is a valid one. So for instance, for the ISR has been shown to underlie many different diseases, including viral infections, cardiovascular disease, metabolic disease, diabetes too, as you mentioned. So in order to say test our hypothesis that modulating the ISR will be disease modifying for, say, diabetes in clinical trials, what we would do is we'd have the typical primary endpoints for diabetes, right? Like weight, blood glucose, etc. But as secondary endpoints, we would also measure, say, blood biomarkers, which might include some of the proteins that Max mentioned, ATF4, CHOP, as a way to see whether or not modulating the ISR in this case is associated with curing diabetes.
Absolutely. It's like a multi-dimensional sort of study question, etc.
Exactly.
Yeah, I like it. It's great.
Thank you.
All right, we've got 4 minutes left here. Anyone else with any comments or questions? If not, then, oh, Stefan, go for it.
Yeah, so I'm kind of still phrasing this question in my head, but so for example, with the example of diabetes, right? The reason that there's cellular stress in those cells that are generating insulin is because they're being asked to make more and more insulin, and 60% of their export or translation capacity goes towards this one protein, and they get overtaxed, and eventually they fail, right? And now you could say, if we modulate the stress response, maybe we can make them more resistant. But you could also hypothesize that if you modulate this, that the stress response is what keeps them alive, what keeps them going over the edge, right? It's a throttle to not overheat, just like your CPU in your MacBook, right? So is there any evidence that you're not really removing the stressor, right? You're modulating the way of dealing with the stress. So is there any evidence that you're not going to get adverse outcomes?
Right. So I think the question there is our biological response to stress optimal, right? And I think the answer is like resoundingly no, because we now live in an environment with the diabetes example where we didn't evolve for that. And then also with things like these chromosomal abnormalities, it's sort of not sort of in the system design, so to speak. So then what are some examples of ways that modulating the stress response actually increases health? Well, it's very clear that things like if we just talk about Down syndrome just to begin with, there are 3 or 4 things, taking minocycline, taking SSRIs, changing BDNF levels, and exercising, that all reduce the memory impairment in trisomy patients, and all of those modulate the integrated stress response. So in the absence of any of those interventions, patients are getting worse. So I think what we're hoping to do is the sort of downside of all those is that they're all sort of indirect modulators. So what we're hoping to do is to directly, titrate things. And again, dynamics might be important here. It might be like cycling on and cycling off in a way that, for example, mimics the positive effects of exercise, creates those hormetic responses, right? There are many reasons to think that our cells are sort of trying to be efficient, right? And so they might not refold all of the proteins, that they could to sort of like save energy, right? And indeed, there are many sort of biological organisms that, show no signs of aging or senescence, period, right? And we like have other kind of evolutionary contingencies that, and programs that cause us to, get old and fail. So I think that there's probably going to be a lot of room to modulate the stress responses, to make health spans longer, and also to modify some of these stress-related diseases.
Yeah,
Yeah.
I don't disagree with that. And we're studying the NRF2 response in Down syndrome, for example, right?
Yeah.
But I think my point was just being that I think you need very specific ways of modulating it rather than abrogating the response. And secondly, some of that might be very cell type specific because neurons and astrocytes rely on each other for energy metabolism, right? So, you might have to do one thing in one cell type and a different thing in another cell type. So that's kind of what I was getting at, that, the interventions that you mentioned as beneficial, I do agree with, but none of them, for example, are what was tested in that, Walter Lab paper, right? With the PKR inhibitor. So the minocycline, the BDNF, the exercise, right? I forgot what the fourth one was. But they do more specific things than just like abrogating that stress response through a PERK inhibitor.
Yeah. Yeah, so in short, we agree with that, Stephen. I think in the end, what we have to do is we have to test different modalities of modulating the stress response, and we have to go after specific indications where modulating it is actually effective, as compared to diabetes, where say to a first order, one just wants to regulate insulin, for instance. But for different diseases such as neurodegeneration, the stress response might be the first order thing. And then again, like figuring out how to should we, increase it? Should we decrease it? And, what kind of downstream effects might either of them have? I think those are all amazing kind of questions that we'll ultimately have to resolve experimentally.
And then the other kind of way we're thinking about this is we've designed some of our screens to deliver us molecules that only work on extremely stressed cells. So in some sense, we're kind of targeting those that are in the most disease state, right? Rather than kind of cell type specific in the respect of function, maybe cell type specific in the respect of sort of disease progression might be another way. Yeah.
Yeah, and on the flip side, we also are sitting on maybe a better analog of ISRIB, which if inhibiting the stress response might also kind of allow us to move in the other direction. So again, like we do have handles basically to play around with.
I think that's a good place to wrap. So this was a great turnout and great engagement. So thank you again, Felix and Max, for joining us. We're currently working behind the scenes on contracting and getting this project underway as soon as we can. So really appreciate the team spirit here, even before we have things that officially signed, and really excited for next steps.
Yeah, thank you everyone for having us. It's a pleasure.
Yeah, great questions. Thank you. Very thought-provoking.
Thanks, everybody. Have a good rest of your day.
Appreciate it.